Dear students, today we will discuss the very first chapter, Reproduction in Organisms. Be ready? First of all, we should discuss its definition. That what is reproduction? Reproduction is an act, or you can say an important biological process by which an organism gives rise to another organism similar to itself. What do you think the mode of reproduction is alike in all living beings? Definitely no. It is quite different in various organisms, but it may be summarized under few heads. Like this, reproduction enables the continuity of the species, generation after generation. It results in formation of genetic variation. This genetic variation is inherited during reproduction. Reproduction involving a single parent is called asexual. Reproduction involving two parents of opposite sexes with the fusion of male and female gametes is called sexual reproduction. First of all, we should make it clear about the asexual reproduction. In asexual reproduction, only a single parent is involved in producing offsprings. As a result, the offspring produced are identical to each another as well as to the parent. Asexual reproduction is common among single-celled organisms and in plants and animals with relatively simple organisms. It is also seen in multicellular organisms. Asexual reproduction in animals. The most common modes of asexual reproduction seen in animals are as follows. Number 1. Fission. This method is observed in the members of Protista and Monera. In fission, the division of the nucleus occurs first, which is then followed by the division of the cytoplasm. Subsequently, the mother cell splits into two equal-sized daughter cells. When the cytoplasmic division occurs in any direction, amoeba, the fission is called simple binary fission. When cytoplasm divides along the transverse axis of the individual, the fission is termed transverse binary fission, as happens in Paramecium and Planaria. When the cytoplasm divides the longitudinal axis of the individual, it is designated as longitudinal binary fission, as happens in Euglena and Warty cells. Binary fission involves mitosis only, and consequently the resultant offsprings are genetically identical to the parent and to each other. In some cases, the nucleus divides several times by amitotic divisions. This leads to the formation of a multiple new copies of nuclei. Cytoplasmic division does not occur during this period. The cytoplasm collects around each nucleus, thus form a single cell. Many unicellular and uninucleated offspring are formed. This method of reproduction is called multiple fission. Example में amoeba and paramecium. You may differentiate it. Number of daughters produced in binary fission divides in two daughters. In multiple fissions, parent divides in many daughters. Time of formation during binary fission, favorable condition, and in case of multiple fission, during unfavorable condition, and fate of parent, nothing is left with parent in case of binary fission, while residual cytoplasm is left in case of multiple fission. Now we should discuss about the sporulation. Sporulation occurs during unfavorable conditions. Organisms like amoeba withdraw their pseudopodia and become round in shape. They create a protective and hard three-layered cyst around themselves. This process is called encystation or encystment. Under favorable conditions, the nucleus of encysted amoeba undergoes multiple division to form large number of amoeba. These structures are called pseudopodiospores. This process is called sporulation. The cyst then ruptures to release all new amoeba in plasmodium. This process occurs at a specific stage in its life cycle. Sporulation may be in case of fungi in covered by a membrane. Next, budding. In this method, cells of some parts of the body of the animals undergo repeated mitotic cell division. This leads to the formation of overgrown regions of cell masses. Such regions of cell masses that result due to mitosis are called bud. A young animal is developed from such a bud. It detaches itself from the parent body and lives as independent animal. Such bud produced on the outside of the body is called exogenous budding. For example, hydra. Enveloped cell mass developed towards the inside of the body are called as internal buds or gammules. Such gammules can be seen in fresh sponge, for example, spongilla, and marine sponge, cycon. Each gammule gives rise to a new animal. This is called endogenous budding. Now, turn of fragmentation. In this method of reproduction, the body becomes fragmented into several or different parts. Each part develops the remaining body parts and becomes a complete animal. This capacity is known as regeneration. As happens in planaria, hydra, starfish, but fragmentation is termed in case of plant like Spirogyra. The vegetative thallus or hypha break up into small segments due to mechanical pressure. Each segment is capable of growing into a new mycelium. This method is observed in some algae like Eulothrix, Didonium, Spirogyra, and Zygnema, and some fungi also like Mucor, Rhizopus, and Saprolegnia. Now we should discuss something about vegetative propagation. In flowering plants, methods of vegetative propagation or reproduction are grouped into natural and artificial methods. Natural methods. In natural methods of propagation, there is development of a new plant from some organ of the mother plant under suitable environmental condition. Such modified organ may develop from stem, leaf, root or even flower. Vegetative reproduction through roots, sweet potato, asparagus and dahlia. Like this, through leaves. In this method, buds develop in the margins of leaves. These buds produce new plants as can be seen in bryophyllum. Through float buds in plants like agave and oxalis, floral buds produce new plants, and in Dioscorea, axillary buds do so. Through stems, like runners observed in lawn grass, Synodon dactylon, offsets found in Pistia, stolons in Nephrolepis, and suckers in mint plants. And now about the artificial methods. Methods are developed for artificial vegetative propagation in which some part of the plant organ is utilized for obtaining a new complete plant. Amongst them, the most common methods are cutting, layering, and grafting. First, cutting. Cut pieces of root when planted in moist soil leads to the artificial inducement and development of adventitious root. New plants are developed in this way in lemon and tamarind. In rose, sugarcane, croton, china rose, and chrysanthemum, plants are developed by cutting that involve stem pieces containing nodes. They are planted in moist soil to develop new plants. Adventitious roots develop from the underground parts of a stem, whereas buds develop and sprout on the aerial parts of stems. The plant so developed is called a cutting. Later, these cuttings are transplanted in proper places. Another method, layering. This method is employed in the cultivation of rose, lemon, grape, hibiscus, and jasmine. The lower branches of the plants are bent and covered with soil in such a way that the tip of the branch remains outside the soil and the middle portion is buried inside the soil. Adventitious root will develop from this buried region of plant stem. At that time, this branch is cut and separated from the parent plant, thus a new plant is obtained. And now we should discuss about the grafting. Grafting is practiced in plants which have difficulty in establishing roots or generally have a weak root system. In this method, two plants of the same or different kinds are joined together. This is achieved by bringing the tissues of the two plants in direct contact with each other. The meristematic tissue of both plants divide and multiply, and eventually the cells of each plant fuse together. The rooted plant is called a stalk plant, and the plant which is being grafted on it is called scion. A plant possessing higher and desirable characters is selected as scion, and the stalk is usually a strong, sturdy and hardy variety. Generally, it applies in mango, apple, pear, citrus fruits, guava, lychee, and many other fruit yielding plants are thus obtained and maintained. Grafting may be of different types, namely bud grafting, side grafting, tongue grafting, wedge grafting, and crown grafting, depending on the methods of uniting the two parts. Significance of the vegetative reproduction. Vegetative reproduction is an ideal method of reproduction in plants where we wish to retain parental traits. Second, it is ideal for plants with less efficient sexual reproduction, small seeds, long dormancy, poor seed viability, etc. can also be multiplied easily through this method. And the third, vegetative reproduction is useful in obtaining disease-free plants. Grafting can be used to bring together the desired character from two plants. Little bit also discuss about the sexual reproduction general properties. Sexual reproduction involves formation of the male and female gametes, either by the same individual or by different individuals of the opposite sex. The gametes then fuse to form the zygote, which develops to form the new organism. It is a complex and slow process as compared to asexual reproduction. As it involves the fusion of male and female gametes, offspring are not identical to the parents or amongst themselves, even with different external morphology, anatomy, and physiology. The sexual mode of reproduction is similar in pattern in plants, animals, and fungi. All organisms undergo general growth before reproductive growth. Only when they are reproductively mature, they can reproduce sexually. This period of general growth is called juvenile phase, and in plants it is known as vegetative phase. The events and processes of sexual reproduction are fundamentally similar in all organisms. However, the structure associated with sexual reproduction are quite different. In all cases, the sexual reproduction is characterized by the fusion of the male and female gametes of the species. For convenience, these sequential events may be studied later. Before the end of this video, we should discuss and keep following points in our minds: that the basic definition and introduction of the reproduction, then various types of asexual reproduction, differences between asexual and sexual reproduction, differences between binary fission and multiple fission. Thank you. Have a nice day.